to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 69th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Warrino, and I have been in the LBA and cell and gene therapy space for close to 30 years now. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I have been in the bioanalytical LCMS space for over 28 years.
John and I are senior scientific advisors here at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS's services. I'm located in Kansas. John is in beautiful upstate New York, and our producer is in the middle of Missouri. So we kind of cover most of the map. We are thrilled to have you listening to the 69th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, then a focus on a topic of our choosing before discussing any feedback you may have had. We're constantly looking for new topics, and we'd be happy to discuss anything that you want us to cover. So again, we're thrilled to have you here, and we are looking forward to a fun episode. To kick us off for the podcast number 69, John is going to go over today's podcast topic a bit before we jump into the news and resources section. John?
Hi, Dominic. Yes, as usual, we'll start with the latest medical research news before going into our main topic, which today is the evolving world of antibody drug conjugates and how KCS can help. We'll talk about the role of ADCs and where they're used in medicine, the ADC Design, and role of bioanalysis in monitoring ADCs and what we can do at KCS.
Yeah, John, exciting topic for sure, but why don't we just dive right in, John? Why don't you kick us off with the first news and resources item?
So first item is small biotech turns CRISPR against bacteria. So obviously we talk a fair amount about CRISPR technologies on the podcast, but this is Snipper Biome, a small Danish biotech, wanted to look at whether CRISPR could be used as an antibiotic, and they're basically aiming to see if it has potential to deal with antibiotic-resistant bacteria. So they derived a CRISPR-based therapeutic from from a bacterium's antiviral system and used it against antibiotic-resistant E. coli in humans. So they did a small test where with 3 different doses and 24 participants out of 36 of the CRISPR, and the remaining 12 getting placebo. And they actually showed that oral dosing over 7 days worked, and with no mild side effects. Patients were able to tolerate it, and it showed that it actually has potential. So it's another step towards applying CRISPR to therapies that could be very helpful in the long term.
Yeah, it's fascinating, isn't it, John? The amount, how clever we are to look at nature and see what it's doing and kind of reverse engineering it, right? That's sort of what this is in my mind. We're looking at how the E. coli, I think, replicate, right? Is that what they kind of took some of their replication proteins, key ones, and then they tried to knock them out, right? And make it into some sort of antibiotic. But unbelievable, John. This is already in phase 1, right? Looked like it's functioning. They got 24 participants, and
yes, phase 1 trial, absolutely.
They don't really tell you too much about it, right? It's hard to discern what sort of E. coli targets they had. But for those that are new to the CRISPR, right, this is technology that there's these repeats that allow you to, it's a phage, basically, a delivery system of a gene. And this is all the rage, all the new latest and greatest in our field. We've talked about it here before, but this is, John, wonderful, right? I mean, we got all these antibiotic resistance, and this is a major breakthrough, John. This is great news. You brighten my day here.
Yeah, just to give you a little bit of the background, it's initially the compound's called SNP-001. Patients with hematological malignancies who are undergoing hematopoietic stem cell transplants are vulnerable to bloodstream infections caused by E. coli. Now, typically you go at that with fluoroquinolone antibiotics, but they are ineffective against antibiotic-resistant bacteria. So it's really coming at these resistant bacteria from different direction, and really, yes, giving these patients some, helping them survive what is obviously, their treatment, etc., or the risk associated with the treatment. So
Yeah.
it says it's a cocktail of 4 different CRISPR-armed phages that target E. coli that resist fluoroquinolone. So they basically, their thought is, in the long term, this cocktail could be used by itself or in combination with fluoroquinolones. So I would think that was if there's other new fluoroquinolones that come on the market that don't have that same resistance. So yeah, it's I thought it was a positive thing to talk about.
It's an unmet need, right? There's all these stem cell
exactly.
therapeutics, but to me, this screams like, there's a lot of people that end up going to the hospital for standard of care. They end up getting some sort of like pneumonia, right? They get what's the other swelling of the brain that happens? Encephalitis, right? Those are the two major. A lot of times, that's what ends up killing you in the hospital, John. Not necessarily your ailment. I think this has potential for that. I mean, obviously, it spawned from the stem cell therapy transplantation field, but I think it can move into that very quickly, and it's awesome. Yeah.
I think there's a lot of potential. Absolutely. We talk a lot about antibiotic resistant bacteria. We know that, in terms of antibiotic investment, pharma companies aren't really looking at it, even though there's a huge concern about resistance to these therapies, because there isn't the money in it. We've seen a number of, over the past few years, antibiotics have been approved, but those companies have still gone out of business because they couldn't make money from their approved product. So to something like this that's approaching from a completely different angle may hopefully give us gives us an approach that can hopefully help deal with this, that what has been a long-term concern, and it's actually a global concern, that antibiotic resistance.
Yeah, no, we can move on, but I mean, John, it's caught me a little bit by surprise that CRISPR were in the antibiotic space. I think it's awesome, and it is going to be something that could really save a lot of lives globally, John, if this can become somewhat of a standard of care. Although we don't know, I think there's still some TBDs when it comes to CRISPRs, but that study looks quite promising, John. And
as said so many times, it's the first step towards going in a direction. Other people will build on this. Eventually, we'll see things that will build on this, and we're all
going to be like in the Matrix. I don't know if you remember that. Just put a USB port right in my head and transfuse me whatever I need. But with that, John,
let's move on. So this is interesting, because as we get older, this becomes nearer and dearer to everyone.
Oh yeah.
This was an assessment by UBS analysts that pickleball could cause $500 million in medical costs in 2023. As someone who played pickleball for the first time in a company get together, was it? I don't know how many months ago it was.
Yeah, about 6 months.
And then aggravated an old Achilles tendon injury. I can attest to this. I didn't cost the medical industry anything, because I didn't go to a doctor about it, and just I just basically nursed it, and it's fine now. But yes, it's like that. It's obviously pickleball is becoming much more popular. It's associated with an aging population.
No, John, it's much less taxing than tennis, right? It's not nearly as hard as tennis.
Well, yeah, but exactly. But you've got people who are picking up and thinking it doesn't seem as taxing as tennis, but then your competitive edge kicks in, so you want to do better, and that's when you strain and pull and do all sorts of stuff to yourself. And yeah, when you look at it that way, over the number of people who are picking up, it could be quite costly in terms of. One analyst noted in a 2021 study of pickleball and tennis-related injuries that estimated there were nearly 34,000 emergency department visits caused by pickleball from 2010 to 2019. Of those, ER visits, 86% occurred in people over 60 years old, and it is mainly strains, sprains, and fractures in the wrist, lower leg, head, and lower trunk. And of course, as it gets more popular, those numbers will only increase. And,
John, if you remember when we played pickleball as a group, I went undefeated. I just have to say that. I just want to know. I think I lost 1 point, in fact, not just any games. But with that said, you weren't the only one who sustained an injury. Remember, there was a couple bodies falling. And there were a couple people that went head over heels, right? So it was. I thought we were going to end up in the emergency room. So this is one of the. So it's a dual-edged sword here, John. You certainly want elderly people to be exercising more.
Sure,
one of the cause effects is this, right? This is like, I don't know. I remember when I first got into the biology field, it was like 1990, right around 91, 92 is when my major was biology. But as I graduated, I remember reading in 94 about how the number one reason for people making emergency room visits was rollerblading. This is the modern rollerblading, John. That's what this is. Yeah, there's injuries here in the heads, and the numbers are staggering. Twice the number of people are playing in 2022. The searching, they even make a note on Google Trends when they search for pickleball courts, is up 110%. I love pickleball, John. My wife and I play. We play with other couples. But it's interesting. I always thought, I wonder if people get hurt doing this. And I hope I didn't jinx myself, but we can move on. It's fascinating how trends in society can impact our healthcare, because 500 million is that seems like, I don't know how much. That doesn't seem like a lot, but I think it's probably underestimated is what I would guess. But that's kind of funny.
It may well be, but I just thought it was an interesting.
I just had to throw in the rollerball too. Rollerblading, rather. It's the modern day rollerblading. I don't know. Still, I think if you looked at emergency room visits, the number one thing is falling off the roof, John. So be careful.
I'm not prone to doing that. Although
I think men at the age of 30 or 20 something to 50 something, the number one, I believe, is emergency room visit is falling off the roof. But
it is actually coming up to gutter cleaning season, so I'll be up on my ladder. But I never do the.
Well, I got new gutters and an 18-year-old that gets up there. So we can move on, John.
Move on. Again, this is another. So this is basically human gene identified that prevents most bird flu viruses moving to people, and there's a lot of discussion in the media about potential for zoonotic viruses jumping from animal species to human, and bird flu has been in the news a lot, particularly in the UK, because with the avian flu has wiped out significant parts of seabird populations around the UK coast. But in actual fact, it was work at the MRC and University of Glasgow Centre for Virus Research found that humans actually have a gene that serves as a barrier against most avian flu viruses. So if the virus can't deal with the BTN3A3 gene, then it's not going to make that jump to humans. So it means that actually spillovers from avian flu to humans is actually relatively rare. Like I say, we do hear about it when they happen, but we actually have an old defense mechanism that actually protects us against a lot of these viruses that are floating around in birds particularly.
Yeah, it's not surprising, John. The immune system is so complex. It's so much redundancy. It's got so many things to combat viruses. Of course, you got symbiotic viruses, you got pathogenic ones, right? We've got tons of viruses circulating through us, some good, some bad. Some are fine in one area. They get to the wrong area. That makes you very sick, right? Same thing with bacteria. It's not surprising at all that we've got some redundancies to help prevent things like bird viruses, bird flu from really kind of wiping us out, so to speak. You're right. There is a periodic jump that can cause some outbreaks, but they're never really that virulent, are they? I say that, but those that are affected with it, but herd immunity really does wipe it out pretty quickly.
I think
they're not as bad as like a COVID outbreak is, right? So they're definitely different there, but wonderful stuff, John.
So just a couple of quick things. I mean, they talk about the H5N1 bird flu was first found in Hong Kong in 1997, and globally since 2003, less than 1,000 people have died. I mean, so there was 873 infections reported, and a little over half of those died. So, I mean, it's obviously not good. But what this work gives you is by if they can test a virus to see if it has if the BTN3A3 gene blocks it, then you're good. If it doesn't block it, it gives you an area, a way you can attack that virus to actually address it. So it does give a mechanism whereby we can, A, filter them out, and B, hone in on actually treating those viruses that do make the jump. So I thought it was a nice little piece of work, and nice to cite Glasgow for a change. So
there you go. Give a little shout out to your hometown. But no, it is an interesting read, John, around just the depth that they went into, right? They go back to like outbreaks in 1918 Spanish flu, right? They look at the 2009 swine flu. I love it that scientists continue to learn from history, so to speak. And, I think as a scientist, you really do have to be a historian almost, John, right? And that's what they did here. And, the H1N1 swine flu, see that one had a bigger impact, but, they talk about bird flu. I can't quite tell if this is applicable to swine flu as well.
I think it's not. I think the difference with birds is that they don't have the BTN3A3 gene, so they don't have any pressure to keep that. So birds don't have it, but humans do, and so that's why birds don't have a resistance to some of these viruses where we do.
Got it. Swine flu was pretty deadly, though, but we can move on. That one actually, that said here, killed 150,000 people. So that was much more where like a bird outbreak might be under 1,000. So I guess the swine, we got to take a lot more. Makes sense too, right? Birds are way further away evolutionary than the pigs are. Anyway, although sometimes you wonder. But good read, John, and more stuff with food, and in general, these are things that can certainly help global health, right? Not just catered to us here in first world nations, but this is definitely something that can have an impact worldwide for sure.
Yep, exactly. So moving on, this is scientists discover contraceptive gene therapy for cats, and this has actually been quite timely because tomorrow we have 3 kittens who are going in to not take advantage of gene therapy, but they're going in to be spayed and neutered. So basically this is some work at the US Cincinnati Zoo and Botanical Garden, and they were looking it's actually, and it's co-developed with a Massachusetts General Hospital researcher, and they were looking. I'm going to have to. They were
so basically like a cat contraceptive that led to some of this, right?
Yeah. So they were looking for. I read through this, and this is
so replace surgical sterilization, right? So in lieu of that, they want to use gene therapy, right? It's interesting, John. Did you as you're looking? Did you say you have 3 new kittens?
Yeah.
What are you? Yes. Are you crazy? What's going? How many cats do you have?
We have 5 at the moment.
You have 5? Is that in? No, so you have 8, or was it 2 plus 3 makes 5?
It's 2 plus 3.
Oh,
I'm 2 plus 3.
Wow. I got 1, and she's a handful. So, John, back to our news here. Sorry, I digress. It's our show, John. We can do whatever we want with it. No,
let's go back to the beginning because I think I have on this. Okay, this is the headline is "Scientists Discover Contraceptive Gene Therapy for Cats," and it was work between a guy called Bill Swanson at US Cincinnati Zoo and Botanical Garden, and David Pepin at Massachusetts General Hospital, and they were actually looking at the anti-Mullerian hormone, or AMH, and they were looking at the effect of that on the ovary, and that was the basis of their research. And when they were looking at it, they saw that how the hormone affected mice. And it was actually suppressing the ovaries. And then they thought, so how could that then be applicable? So they then looked at cats, and cats are obviously, there's an overpopulation issue with cats. Certainly, there's concern with cats and their effect on the natural environment, because they're killers, and they like being out there, decimating the bird and whatever population, the number of different species of animals we've had through this house, thanks to cats. So yeah, it's like, what can they do? This was a potential approach to look at how could they, because of the ovary suppressing effect of AMH, could this be used as a contraceptive in cats? And this is to get away from the spaying and neutering approach, which is quite an invasive surgery for cats. And as I said outside of this podcast, we actually have 3 kittens who are, tomorrow will be going to the vet to be spayed and neutered. So they go in overnight, and obviously the female she stays overnight, and it's quite invasive for the female cat. So they administer it to a limited number of cats, and what they found is this gene therapy has actually it has prevented cats getting pregnant. And they did controlled experiments where they put females who'd been treated with the therapy, others who hadn't, in with males, and all the ones who had been treated, they got no pregnancy over an extended period of time. So there's potential here, and these cats have remained. They haven't got pregnant over, I think, quite an extended set of years. They've got one group they've been screening for 2 years, and then one of the guy in Cincinnati has had cats for, I think, almost a decade, and they haven't got pregnant in that time either. So I'm going to pass it back to you.
Yeah, it's fascinating. At least 2 years is what, but maybe it's up to 10. But 2 years efficacy, John. It's really a great story here, right? They were actually studying ovarian cancer, right? They were looking at this anti-Mullerian hormone to help treat ovarian cancer. And just often, as science does, when you're looking at one thing, you suddenly realize, wow, I think I can fix something else over here. And that's what led to them realizing this could be a contraceptive, right? Yep. So that's a great story. It's kind of like how we've come with things like, cardiac medications have come a long way, John, is what I'm trying to say. A lot of your meds, we found out, had male enhancement to them. So
sure. Absolutely. Yeah.
This is a similar sort of story, right? They were chasing ovarian cancer, and next thing you know, they realized they could have and it's a serious problem, John. Not only is spaying a cat, it's not so much, it's invasive, it's costly, but it's just, there's so many feral cats or
absolutely,
wild cats that need this, and this could be a tremendous way to help control the cat population.
That's exactly it. I mean, it's less about the domestic cat than, like you say, all the strays. And if you've got something like that depends on administering an injection, and you've got them under control, that's a big deal.
Yeah, no, you're right. Some of them, and as I look here, some are now 10, 11 years of age, and no problems. I think it's wonderful, John. I wonder.
So basically, just to give a little bit of background, the researchers designed an adeno-associated virus vector using the genetic sequence that encodes feline AMH. They then injected it into thigh muscles of 3 groups of 3 female adult cats, give them either a low dose of therapy, a high dose, or an empty AAV9 that served as a control. The transgene prompted the muscle cells to express AMH, which then made its way through the circulation, ultimately to the ovaries. And they inject into muscle because the vector can easily infiltrate the muscle, and it lasts a long time with that approach. So,
yeah, John, I'm having like such a giddy moment here. One shot did all that, right? Like one shot. And they have what appears to be protection or contraception, preventing pregnancy for a decade.
Yes, exactly.
Amazing. Yeah. The AAV technologies are just unbelievable, John. So proud to be supporting them in the clinic, and who knows, the sky's the limit for this, as well as, like we mentioned, CRISPR. But these are just incredible modalities, John, that when we started, science didn't even really exist, or were in their infancy. And here we are today showing incredible advancements for animal health in this instance, but in general world health. So just fantastic story here, John.
So just a final footnote, because of the potential here, they Pepin, who is in Massachusetts, he has co-founded Oviva Therapeutics to that obviously take this further. So that will be an interesting one to watch in the longer term.
It's really cool. What do we got, John? How are we doing on time? Yeah, we got
last
time for a
one.
couple more.
Last one, and this actually will be quite brief, but is also really exciting. It's a couple. It's talking about two companies. So I'm going to throw out both headlines. In the first, FDA clears a type 1 diabetes cell therapy from small Chicago biotech, and Vertex's cell therapy gives early hope for weaning some type 1 diabetes patients from insulin. So the first one is a company called Celltrans, so a pancreatic islet cell therapy, which takes pancreatic cells from deceased donors, and this is it's a cell therapy for patients who are unable to approach average blood glucose levels because of low blood sugar, even after intensive management. And what they found for in the trials is that longer term, a good proportion of patients actually didn't need insulin administration, and the Vertex cell therapy is it's obviously it's behind the Celltrans in terms of where it is on the development scale, but it's similar deal where it's showing effectiveness in small patient trials where some of them are becoming insulin free. So in terms of hope for type 1 diabetes patients, this is obviously an approach with huge potential.
Yeah, amazing. This is an adoptive therapy. Well, xenotherapy, I guess, but really an adoptive therapy. These are cells from deceased donors, right? So these are true pancreatic cells that they perfuse right into the liver portal vein, John. This is great. And then these patients that had never had normal glucose levels do. Unbelievable, John. That is just an absolutely amazing therapy in my mind, because I don't know if you know much about pancreatic islets. They're very fragile, very mesh-like. So I'm fascinated by how they harvest them and how they put them into the donor. But I mean, my goodness, I can't believe how effective this is, John. That just seems crazy to me.
So basically, yeah, the approval of the Celltrans therapy was based on 2 single-arm studies comprising a total of 30 patients who received between 1 and 3 infusions. Across the 2 studies, investigators found that 21 patients didn't take insulin for at least a year, 10 didn't need insulin for more than 5 years, and 5 patients had 0 days of insulin. 5 patients had 0 days of insulin independence. So it isn't obviously applicable to everyone, but when you've got a reasonable proportion for more than 5 years, that's it's really hopeful.
Yeah, I mean, if you're taking insulin daily a couple times and suddenly you get this one time, and some patients were given a second one, and you can go a year. I mean, that is such that has to be so great. And some of them are up to 5 years. I mean, man, this is great stuff here, John. I'm sure it's for it's an insulin, it's a diabetic type 1 diabetes is what they're treating here.
Yes.
So it's a little bit different than the type 2, obviously different than type 2, but as somebody who's got a niece who's a type 1 diabetic, I want to send her this article.
Yes.
God, she wears a pump and everything, and it's a pain, and poor thing. And I don't know if this is an option for her, but certainly I know if I were sitting there having to wear something or pump my daily injections, or even better yet, having to wear something on my side or arm, this sounds a lot more a lot of freedom. She's only 20, so I think she'd be a great candidate. But I digress. John, what about this Vertex one? Why don't we jump into that? But John, what a great story. I'd love to know the procedure for how they get them from the cadavers, and how the whole thing is fascinating to me. But they don't go into that detail.
I think this is going to be a watch this space, because there's a number of other companies are looking at it too.
Diabetes is a big space for sure.
Well, it's like when you're talking about how looking at one thing leads you down another space. I mean, we talk diabetes, and obviously the big news for type 2 are the GLP-1 analogs, but now
Yeah.
there's even more focus on them in terms of potential weight loss.
Yeah, they're seeing some
Uh,
side effects though, right? I think
yeah,
they're.
oh, absolutely, yeah.
Yeah.
As with any drug, but it's interesting how something's developed for one application, but just by observing the clinical outcomes, you may actually open up to a bigger
Well, that's a
market or
bigger discussion.
another
Yeah, I think
another
that's like a
clinical
whole
unmet need.
podcast in there,
Yes,
John, right?
absolutely.
Like the GLP-1, because it's fascinating to me that like you don't want to put the time in and sacrifice in just to eat better and exercise. That's
Mhm.
coming from a guy who's a big dude, right? I'm always battling my weight, but I have no desire to take the medication just from
No.
unless
Well,
I don't know. Well, I digress, but for me,
we digress very
yeah,
quickly, and I'm still going to say my one piece is one thing that put me off. It was I read an article in the Guardian, and it was saying it basically put the person off food, and that's partly
yeah,
why partly where some of the weight loss came from. And it's like I love food,
yeah.
that's I mean, to me, food is fascinating. Seeing what skilled chefs and people can
Yeah.
do with food is amazing. To take that joy out of my life is something I'm definitely
Yeah,
not
well,
interested
so to your point, I know somebody on it. A good friend of mine has taken it, and when we're out, he's like, he's like, I'm not hungry, but he's like, I am, but I can't eat. It's like a weird. He's often nauseous. It took a while to get the dose right. Then when
in.
he was on a good dose, that started not to work as much, and so he's always kind of adjusting things. But there's just times when like he'll eat a couple bites of something, and you can just see he's like nauseous. And I'm
Yeah,
like,
yeah.
just to your point, I'm like, that seems I don't know, man. Life is short, man. I want to enjoy that hamburger. But anyhow,
Yes, let's move on to
yeah,
the main topic.
yeah.
And so we thought, actually, I honestly thought we talked about this much more recently than we had.
Nope.
The last time we discussed anything to do with ADCs, it was in amongst other topics, and it was actually about a year ago. So it's a good time to revisit ADCs and what's happening in that field. And so basically, our topic today is the evolving world of antibody drug conjugates and how KCS can help. And starting off with, what are antibody drug conjugates, and we're going to call them ADCs from now onwards, just to make our lives much easier. They've really shown their to date they've really shown their potential in cancer treatment. So it's what you're doing is you're linking a potent small molecule payload in some of the approved medications. These are actual toxic payloads, and then they're attached via linker to an antibody that will then connect to proteins on the surface of tumor cells. So you're actually directing that toxin to the cell where it can be medically effective. The reason for taking that, you couldn't the payloads you couldn't administer by themselves because the effect on the whole body would just be really it'd be devastating, and that's part of the what where that's part of why ADCs, why people are so interested in them, is by having this very directed approach of delivering the agent to the tumor. You're trying to avoid the systemic effects that we have from a more broad-based chemical therapy that a lot of oncology patients deal with. So it's really trying to get around that type of issue.
Yeah, it takes the best of the monoclonal antibody space. Those often described as guided missiles, right? That's a guided missile approach. And then, as you mentioned, you could take the chemotherapeutic and you can use a much lower, far less toxic dose of it because it's basically being the payload is what we call that, right? It's
Yeah.
piggybacked onto the biologic and a lot of, and we'll talk a little bit more about the designs, but that payload does not fall off as it makes its way through circulation, and as the guided missile makes its way to any tumor or wherever it's trying to target, as it binds, that's when it will deposit. So you're right, you don't get hair loss, all the attributes, all the bad things that come with small molecule chemotherapeutics. And not only is it side of it, John, in theory, it's much more effective if it makes it to the site
Sure.
too, right? So
Yes.
that's the part of it.
Yes.
But before we dive into the whole design, we talk about anti-drug conjugates, and oftentimes they're monoclonal antibodies, but they can be other things, right, John? A lot of people call them anti-protein conjugates or anti-therapeutic conjugates. This is a very diverse field, but
Yeah, I think
we can just call it as any sort of protein, oligo, any sort of therapeutic that has some sort of payload. And the payload doesn't have to be a chemotherapeutic either. It can be some other type of therapeutic as well. So it's a very diverse field, but I do think the ones that are on the market are traditionally
that's
monoclonal antibodies with some sort
Yeah.
of SN38 or MME traditional chemotherapeutics on them.
And that's exactly it. Right now, the approved medications are the ones that really fit that mold of antibody linker toxic payload. Certainly, in terms of people we're talking to, there is a lot more discussion about what can we link, what can we use as the targeting mechanism. What can we use in terms of linkers, and what can we use as different payloads? And yeah, we certainly will touch on that during
let's just jump right in, John. Right, so we kind of went over a little bit of what they are, or what maybe do we touch on the unmet medical needs, John?
Right?
Right? This comes into.
Sure. Yeah. I mean, we can go wherever you want. I mean, we can talk design if you want. I mean,
let's talk about the design.
Because basically, you're looking, like say, we're talking antibody, payload, and linker are the big deals. What we're looking for with the antibody is low immunogenicity and high affinity for a particular site on the tumor, and that will then help your efficient delivery to the site of the tumor, and hopefully your effectiveness of once you have the payload there. You also want to have a longer circulating half-life, so it spends some residence time in getting there.
Yeah, John, I like the idea of just how it takes the two major. I know we talk about cell and gene therapies, and that's an emerging modality, but chemotherapeutics been around 40 plus years. Biologics is now entering 20 plus years. The two of them coming together is just a fascinating evolution of science. It's become, it's been a hot topic for a decade, but really in the last, let's say, 3 years, right, has there been major breakthroughs in treating solid tumors. Let's call it what it really is treating, is solid tumors. And we're blessed to be supporting one of the best, one of the number one ADCs on the market, as well as what John, I think 9 or 10 other ADCs we help support, right? So we do quite a bit of this work. And the technology is, the monoclonal has a lot of engineering and technology, or whatever the framework is, right? It doesn't have to be a monoclonal, but whatever that framework is, right? Whatever is the guided missile, so to speak. The cytotoxic payloads are fascinating, John. We could spend a whole podcast talking about each of those components and how they're engineered and the evolution of it. But those payloads are, instead, the traditional chemotherapeutics that you might give somebody is you give them, it's a potency that's very low, but you give them such a large bolus of it, where this is a highly potent warhead, but such a small amount, right? Like maybe 1 or 2 DARs, right? The drug antibody ratio. But really the technology is what? The linker, John.
Yep. Yeah, absolutely.
And then the other designs, and we'll talk more about it, but that linker, so how it's conjugated is really is a very diverse portion of it. And that's when we talk to ADC companies, they get very touchy about that, don't they? They don't want to reveal what's happening.
Oh, sure. Yes.
That is the technology. Some are cleavable, some are non-cleavable. And then really, in my mind, that linker, some of the best ADCs out there have linker technology that's, what's the word? Not as advanced. And that's I don't know if what I said made sense, John, but like the ones that are, some of the ADCs that have a very strict regimen of how they're made. There's 2 payloads, they're in a cysteine, they're cleavable, blah, blah. They're not always as effective as those that, like, some clients are like, yeah, we think the DAR is like 8, it falls off here and there, and those are highly effective. So it's really, to me, it's a fascinating field, because sometimes somebody will roll out an ADC and we're like, wow, this is really, this seems to be really put together. They know what and it's not really working that well. And then others are kind of like, yeah, we don't really know how we made this, but it's highly effective, which is funny. And even some monoclonal antibodies are that way, but I digress. There's some really good, like cetuximab. People trying to make a biosimilar of cetuximab are really struggling. So anyhow, John, that's my 2 cents, and I'll let you jump back in. But that linker is really where proprietary pieces lie. And then we can talk more about the next wave. But when you think about the 3 building blocks, it's the framework, often a monoclonal antibody, the payload, often chemotherapeutic such as SN38 or MME, and then some sort of linker. And that linker conjugate becomes very tricky bioanalytically, doesn't it?
Sure. And the linker is critical to the whole success. I mean, you were mentioning it needs to be sufficiently stable that you don't release the payload from administration site to target. You want to keep that to minimum, but then once you get to the target, you want the linker to operate such that the payload is released quickly to address the target. You were talking about different linker designs. 2 of the ADCs, Trodelvians and Lonta, both actually have PEG within their linker, and that's how they extend the half-life of the ADC. And that's obviously the longer the half-life, the more time it has to interact with the tumor site, etc. So that's how critical that the linker design can be. It all plays into that. It's funny when you talk about DAR ratios. I was just like reading while you were talking, and yeah, we typically deal with ADCs where it's a DAR of 2, but some of these other ones where you've got a much higher DAR, say 7, 8, you can actually have more off-target side effects because you get like a spillage effect. So there's obviously a balancing act in terms of how high a DAR you want to have, depending on the target you're trying to address.
Yeah, John. Yeah, it's definitely big science there, trying to design a we keep mentioning that DAR, that's the drug to antibody ratio, meaning, do I want 2? Do I want 6? Do I want 8? Do I want them cleavable? Do I want them non-cleavable? And then, like, in theory, it's like, well, 8 is better than 2. Well, not necessarily, right? If I put 2 highly potent payloads on there, that could be improved. So, John, we as we work through our agenda here, we talked a little bit about the design, right? What else do you want to talk about the ADC design? Maybe this is a good point to talk about. What is that next evolution of the design, John? And where I'm going. Or do you have more on the design of it? But
I don't think there's much to. I mean, we could. I haven't got much to talk about. We could go to that in a couple of minutes. One thing we haven't talked about is the mechanism of action. So there's a 5-step. Because again, let's just touch on this. So basically, there's 5 steps in terms of action. The ADC binds to the antigen on the target cell. The ADC antigen complex is internalized by endocytosis. That's step 2. 3, the ADC degrades in the lysosome. 4, the cytotoxic payload drug release and function. And then 5 is target cell apoptosis. So I throw that out as the 5 steps if you want to talk through that, and then I want to talk on what is actually approved and the kind of payloads they have.
Yeah, so the John, you kind of nailed it. You used a lot of big words in there, but basically it's that guided missile making its way. And I always think of it as like a Brownian movement, right? It's just it's given systemically. It makes its way through your vascular system, and then it can find its target. And a lot of it is just flushed out through your liver or your lungs or other areas. But in theory or in practice, somewhere in there, enough of those little guided missiles or antibodies is and typically what we're talking about. They will make their way to the tumor site, right? And then, as you said, so John, that's the power of it, because some monoclonal antibodies are therapies in and of themselves. They don't need the payload, because that if you start littering a tumor cell or some sort of solid tumor with antibodies, that FC portion will attract the immune system, and that'll trigger an adaptive immune response. So monoclonal therapies in and of themselves have had some therapeutic efficacy, sure, what I just described. However, they're often in the tumor microenvironment, they're just not that effective, because the tumor microenvironment is like, it's like the surface of the sun, in terms of our, sure, It has a lot of volatility to it. It's going to have the tumor. And remember, that's just you. It's what's happened is your cells, your endothelial, epithelial, whatever it might be, mesenchymal, they've been corrupted in such a way that they're producing natural cytokines, chemokines, but they've been augmented. They're producing things to diminish the immune system. They're producing things to suck up chemokines or chemoattractants and things that want to attract immune cells. They're producing, some of them are virally infected. I think all tumors are virally infected. Don't get me started, John. Maybe not the causative, but they're going to have decoy proteins up there to prevent your innate immune system from recognizing it. They put certain MHCs or TCR. They put things to avoid adaptive immune responses. So it is like the surface of the sun. So that endocytosis process is still somewhat hard to overcome. Well, it doesn't have to if the payload is there.
Yes,
you know
exactly.
What I'm saying? So then if you've got, think about it, if you've got a combination of the antibody itself kind of pissing the tumor off, for lack of a better word, aggravating it, and then you dump in a couple payloads, man, that's why it's so effective. So we don't know if it's one ADC that has to be there, 10 million ADCs, and I say one, obviously there's Avogadro's number, 10 to the 23rd per mg or something. It's a huge amount of antibody in a very small amount. So, but that's not studied, John. Nobody knows how many ADCs need to bind to a tumor to kill it. That's not, and if you know that article, John, send it along. But that's the power of these things. It's the combination of the the biologic having therapeutic effect through what I just described, as well as the small molecule sitting there dumping itself. And John, in theory, they want the entire thing to be internalized. So if it does, so one effect is it can trigger an adaptive immune response, but most of the time it's just going to be gobbled up. And if a biologics gets gobbled up and internalized, it just goes to the lysosome and gets broken down. Well, if you put a chemotherapeutic in there, John, guess what? You just killed that cell. So that's the power of this. It has a couple different mechanisms of action to it. I hope I drilled down a little bit to cover it, but those are the whole sorts of, in addition to the biologic itself has properties, but that small molecule, what they really want. So why monoclonals aren't effective is because they get endocytosed. And they don't have the ability to trigger innate and adaptive immune responses.
Sure. Yeah.
But in this case, that's wanted. So the very thing that was inhibiting monoclonal antibodies, you're not taking advantage of, right? By having that payload on there. And it's John, I hope you can hear it in my voice. This is fascinating stuff to me. And I love the idea of like, the technologies, and maybe we'll drill in a little bit there.
Just to jump in, I mean, right to date we have 13 ADCs approved, although one was withdrawn. I assume probably because of side effects, but it's a growing list of obviously approvals. You see the mix of payloads are there's some like natural compound analogues, so calicamycin compound class, the monomethyl auristatin E is a common payload, MMAE, which we'd refer to. MMAF has been used, so another analog to the MMAE. SN38, which is actually an active metabolite of irinotecan, which is another small molecule prodrug for oncology. So that's where you're actually using the administered drug to get to the site and then release its metabolite that is then attacks the tumor. So there are alternative approaches there. And then more recently, we're seeing actually more proprietary payloads, such as I think Enhertu, which is Enhertu is another camptothecin analog. Enhertu is a lot. There's a lot of buzz about that. It's Daiichi and AstraZeneca developed ADC, but then when I was unaware of is Lumoxiti, which is another AstraZeneca, actually has a Pseudomonas exotoxin A fragment, which is a 38 kilodalton fragment. So there's your first approved one. It's actually a step away from what we're calling small molecule payloads. So I didn't know that until I was doing some background research on this. So that's where now we can talk about different designs of
before we move off of that's. Yeah, John, that's interesting. A Pseudomonas toxin, right? Why not poison ivy? I've always believed that poison ivy antigens would make great therapeutics, but I digress, John.
There you go.
No, but it is fascinating, John. Your list.
I mean,
here,
it does beg the question. I mean, we know there's obviously there's a lot of what we work with are derived from natural products or are based on natural products, and they've been fine tuned from there. You know, we come back to GLP-1 analogs, which aren't part of this discussion, but they were based on a peptide in the Gila monster's saliva,
yeah.
and so it's.
Yeah.
That's where some of that's where ideas come from. Nature solves some of this stuff for us. We just have to figure out how to make it work better in a human environment.
Just to wrap this up, there are is it 29 approved? The first one back to 2000, right? So it looks like there was
it's 13 approved.
30 or 13?
13.
Oh, okay, I see. And you've got a list of other up and coming. So it's 13 approved.
So I basically listed what were approved and then what the payloads were, because that to me was the interesting piece.
So 13, and really it looks like one in 2000, but then 2017 it was either reapproved. That's the Pfizer Wyeth. Man, I haven't heard the name Wyeth in a long time. Same with Millennium's on here. It's Takeda, but John, you can see the explosions really happened in the last. It's like 2019. Over half of them have been approved.
Yeah, absolutely. Yes. And we know that from the number of people we're talking to about possibly working with ADCs, etc. Yeah. I mean, because bioanalytical is a huge part of that. I mean, we could maybe touch on bioanalytical and then go to further designs. I mean, because
let's talk about the design real fast, and then we can go into the bioanalysis of it.
Yeah, not a problem.
Yeah, John, the latest and greatest is twofold, right? So they're getting very sophisticated ways to unleash the payload, right? And that's where a second small molecule is dosed to release it. We're very familiar with that. That's fascinating. I don't know if you have anything you want to add there, but I think that's an incredible technology that they'll systematically give the ADC, and then what do? They give some sort of Lamisil, I believe, can be used, some sort of very benign small molecule to release the payload, right? Isn't that what causes the cleavage?
It's what's called the chemical trigger.
There you go.
Then basically, once you're in the target, once you're at the target, the trigger helps then break the molecule to then do its to basically have go to work and do what it needs to do. So I basically picked this up from someone and said recent advances in ADCs include site-selective attachment using engineered antibodies, site-selective modification of native antibodies, advances in linker chemistry, which comes back to what we were saying earlier. There's an awful lot of discussion on that, and we're seeing that in some of the candidates that come over our desks. It's obviously looking at different payloads. We talked about the mix, even of the approved ones, between the traditional, like the SN38, like the MMAE, but other payloads are being looked at. There's different linkers, the triggers that you touched on, and it's just more designing strategies, and so there's a lot of focus and all that. And then actually want to touch. I mean, we've heard discussion about antibody oligonucleotide conjugates, because it's looking at what the oligonucleotides are, almost like a special type of therapeutic, and so it's looking at, can we target oligonucleotides to work on site, because oligonucleotides have got a lot of more application in terms of addressing unmet medical needs. I remember, when I was working early on in bioanalysis, we never really talked about oligonucleotides, but you heard whispers of a couple of companies who were working on them, mainly being who are now Ionis and then Alnilam. But it was just like there's these companies working on these compounds, but we weren't sure quite what they were going to do. And then more recently, there's been an increasing number of oligonucleotides approved, and much more focus on what they can possibly do in terms of helping patients. So attaching those to antibodies is of huge interest to pharma companies.
Yeah, John, the topic of oligos is fascinating, and you touched on two major players in the space, but oligos have some advantages over monoclonals and they're one of just the sheer size of them is one of them. I always think of oligos as having like hormonal like properties to them. But anyhow, we could probably just maybe it should be its own podcast. Before we and you know they are the future certainly with in terms of conjugation. So again, anti-therapeutic conjugates is what that kind of falls into that area. But before we move off of some of the advancements,
yeah, I'm just going to just touch on the oligos if you don't mind, because
sure.
So basically, problem with the oligos is cell uptake internalization still represents the biggest hurdle towards successful oligonucleotide therapeutics. Their intake is really hindered by the polyionic backbone, because they've got a very negative backbone, and that gets in the way of their uptake, and just their size. So it's that's why it's they're actually think part of the research is looking at antibody AOCs is actually combining the antibody with fragments of the oligonucleotide. So you're actually trying to get smaller pieces of the oligonucleotide into the target. And working from there. So there's, like I said, there's a lot of discussion on this.
Yeah, no, they're fascinating. I still favor the MAB, but that's because that's my comfort level, John. As you mentioned, oligos have always been a whisper, although in the last, I'd say 3 years, we do a lot of oligo analysis. Obviously, they're very powerful. A lot more. Those oligos have a lot more. I think of them as activity than I would have thought. They're very active, right? They have a lot of functionality to them, and the uptake of it is different because it has to be passively uptaked, right? Like cells just constantly sample the environment, and that's how it gets taken up. Where an antibody, it binds to the surface of the cell, it's going to trigger it to be taken up. We mentioned clathrin-coated pits as being one of the ways. All sorts of cell trafficking that can take it up. But John, I wanted to maybe just take one more. I know we talked about oligos, yep. But when it comes to the advancements in ADCs, I know you listed a whole bunch of things, and I think they're fascinating. But one of the ones we didn't touch on is these like, I think of them as like pro ADCs, and so people are putting a little hinge onto the antibody to block the FAB binding of the cell. So of the antibody, sorry. So meaning, antibodies have a bivalency to them. They've got two arms, right? They've got these two FABs to it, and they'll bind to a target. And oftentimes, if it's just one of them, it's some steric hindrances. But what are they doing? They're blocking that, and then they're putting a little peptide over it or some sort of protector of the binding region of the antibody, and then what causes it to be released? It's the tumor microenvironment, John. So that that antibody it'll avoid any cross reactivity. It won't bind to normal tissue, right? So that antibody should be that guided missile gets enhanced, and the only place that it will actually bind is if there's certain, and it's really easy. Tumors secrete all sorts of peptides that other tissue don't. They're constantly having to destroy angiogenesis, right? They have a lot of angiogenesis process. They need a lot of vasculature. So because scientists, we know that, we take advantage of it, and I can't tell enough about. If as a scientist, you should be looking backwards, that reverse engineering. And it's so fascinating how they put that together to say, hey, let's create this like pro ADC.
Yes, so absolutely.
Until it gets into the microenvironment, it's not going to bind. In theory, it shouldn't bind, so it could circulate longer, and then we can talk about extending the half-lives and all those sorts of things. But those types of things, the combination of using a small molecule to release it and the combination of using the tumor microenvironment to open up the binding region of the FAB, just unbelievable science to me, John. Like, I think those are Nobel Prize winning thoughts in my mind, but maybe I'm just a little bit of flamboyant when it comes to this type of stuff. So, John, this has been a wonderful discussion. I think we covered all of our topics. Maybe a little bit on bispecifics, and there's some powerfulness there if you want to talk about it. But
we just say bioanalysis. I mean,
that's where I was going next. I really wanted to talk about that we got two more topics before we wrap up, and that is like the role of bioanalysis in monitoring ADC. So I'll let you go first because it's a little more complicated on the LBA side, right? But maybe you talk about the roles and just in general, and I'll fill us in around that
Well,
immunogenicity component.
I think to some extent because the field's evolving, the bioanalysis is evolving, and if you look traditionally, if we sort of go back, to some of the ADCs that have been in the market, obviously that you're talking those traditional small molecule payloads. So really, there's I think there's four different. If we're looking at what the drugs do in the body as part of a pharmacokinetic profiling, you want to look at total antibody, total ADC, which is with the payload attached. May look at total payload, and you're very likely look at free payload. And free payload is a measure of the payload that has come off in free circulation, so it's not at the site of action. So that's obviously something you want to keep down as low as possible.
So John, you described 4 methods. So from a bioanalytical perspective, you need a minimum of 3 and oftentimes 4 bioanalytical PK methods, right? You mentioned it, the free payload, the total payload, and those are often done by LCMS. And what we mean by that is, does that payload come off or is it on, right? We have a variety of ways to measure that, but it's all LCMS based typically. Then you mentioned the total antibody. That's the minimum, right, John? Sometimes you can get away with just those and do some ratios to tell you what the total ADC is, right? So just that the total antibody is a measurement oftentimes done by ligand binding assay. It can be done by mass spec. We do a lot of mass spec for all of them. But it's often a ligand binding assay, and it's just measuring the monoclonal antibody. Doesn't matter if the payload's on there. Doesn't whether it is with it. It's called nakeds. Oftentimes they call it a naked, antibody. Whether the payload's on there or not, it isn't discriminated within the assay. And then that total ADC is where I think as a company we excel. I think that's KCS's sweet spot because we can tackle that total ADC by 2 ways, right? One, there's been a lot of advancements in antibodies, commercially available antibodies against the payload. So you can use a ligand binding assay to measure total ADC because you come back with an you use a sandwich ELISA or a MSD assay, and you would you capture with the anti-payload, and you can come back, right? And you can come back with a detector that's against the monoclonal antibody. You can't use the converse because that would pick up every antibody, but so it needs to be a very specific anti-payload. Now, they've come a long way, but they're very expensive, aren't they? The alternative would be to do it by hybrid. And before we get into that, the hybrid approach is to capture against the small molecule and then look on it by mass spec. And we have a lot of generic methods for monoclonal antibodies. We have those off the shelf and ready to go. So, John, I don't know if we've covered all 4 PK methods. Really, it can be 3 or 4. I think that's still, to me, I think the FDA is really looking at you to do all 4 these days because of the advancements I've made. But we have some legacy assays that don't look at the total ADC, right?
I think I haven't been in discussion with the FDA, but certainly we're going to be looking at all or some of those assays to get a good picture of what's happening in the biological system.
Yeah, and maybe this is a good time to dive into the LBA versus hybrid, and then I can touch on the immunogenicity component. But John, that is really what separates us. A lot of companies can say they can do it by both, but we've been doing it for 5+ years, and maybe others have been doing it longer, but it's all under one roof, John. That's the power of what we do. We're able to, whatever information, say you've got a method that's ligand binding assay based, we can take that and adapt, we can push it through as an LBA method, but we also have the ability to say, hey, we could look at it by an affinity capture followed by mass spec, or that so-called hybrid mass spec. And oftentimes, non-clinically, the method may work, but clinically it won't because of, it doesn't have the specificity. You're using some generic or commercially available reagents. You're taking advantage of trying to detect a human antibody in a non-human species. But when you flip it, it becomes when I say flip it, when you go into the clinic, those reagents are no longer viable sometimes, right? That's where the mass spec can become a very powerful tool. If you have a good anti-payload antibody, you can use that as your method for pulling out your total antibody. Your total ADC. You're always going to have to invest in a monoclonal antibody that's an anti-id. You're going to need an anti-id for the total antibody assay, whether you do it by mass spec or hybrid. It's not going to matter. John, I hope I kind of touched on it. For us,
I think for us it's important just to touch, just having those two technologies available means we can readily flip from one to the other if one shows an issue. I mean, you're right. Traditionally, we've gone with LBA for the large part of the molecule, and I think when we talk to customers. To begin with, we still will largely be thinking that direction. But if we see issues with the LBA assay, we have that ability to flip.
I would agree. For total antibody, most clients they're comfortable with the LBA.
Yes,
we don't want to reinvent the wheel. Well, that really dictates it a lot, is what's your comfort level.
Yeah.
But if the total antibody, if you're looking to do a total ADC, they're very different, right? Total antibody is how much of the mAb is in there. Total ADC is the total drug, right? That method by LBA, I feel like, is a struggle. And it's not just KCAS, that's an industry-wide thing. We're looking at total ADC by mass spec, in my opinion, is going to be a much more robust and a much more validatable method if you have the right building blocks, right? So, I mean, if you get good building blocks, you can do it by either. But in my opinion, you need only one antibody that's good on the hybrid mass spec side of things.
Yep.
For that total ADC. It's just John, we've got programs where the entire PK profile is done by mass spec. And then we have others where just the free payload and the total payload, and then the rest of it's done by LBA. But if someone came to us today and wanted our opinion, John, I think it'd be an interesting conversation. We'd have to learn more about the antibody, but obviously the mass spec pieces of it that's a no-brainer. To me, if you have a good total antibody by ligand binding assay, I'd do it that way. If you want a total ADC, if you don't already have one established, we're going to recommend hybrid. At least that's my opinion, John.
Okay. Yeah.
Because oftentimes, non-clinically, people don't worry about the total ADC as much. It's when you get into the clinic that you really want to start making that investment in what is the total ADC in circulation. So I hope we did a good job of broaching the subject. I know Dawn Dufield is somewhere listening to us, cringing, wishing she was here, but we'll bring her on next time, John, in the future. Maybe in a couple months, we'll have a reboot on this. But
sure, absolutely.
John, any last comments, and then I'll touch on the immunogenicity of ADC.
No, I don't. Let's move into that piece, and then we can wrap up.
Yeah, I love a good immunogenicity conversation. So in this case, I take Occam's razor. The path of the simplest solution is the best solution, and so just start out with an anti-drug antibody assay, very traditional to any sort of mAb. That's going to be what I would do non-clinically. There's been a lot of changes in the non-clinical regs. ADCs kind of fall into a grayish area, in my opinion, on the immunogenicity risk assessment. To say they are low risk is just not right. They're probably in a medium risk because of the payload, right? There's a lot of stuff going on there.
Yeah.
So, whether you need to validate them is, at least qualify. I would qualify a 2-tier method. And what I mean by that is some sort of screening and titer. That's our preference in the non-clinical space. I would, even if it's a mAb or a total ADC, I'd take that approach unless I'm seeing something really toxic in my ADC. And even then I'd probably re-engineer my ADC versus investing heavily in an immunogenicity non-clinical assay. So non-clinically, you can get away with qualifying the method, and in my opinion, all you need is just the total ADC. That's it. Sometimes you can just use the mAb. In fact, some do a total, just the actual guided missile or the framework or however you want to call it, but the total ADC versus the total antibody doesn't really have a big impact non-clinically. You can develop a method against both. Obviously, the preference is to use the actual drug. But as you move into the clinic, that method, that's when you have to get into looking at what we call domain specificity. So you're going to still develop a primary method that in this instance is against the total ADC, and it would be a standard MSD bridging assay, and you're looking at any sort of that should be able to bind that ADC bridging assay, excuse me, the MSD bridging assay for ADAs takes advantage of the drug, and you biotinylate that and ruthenium label it, and then the bridge is formed by having an antibody that binds to it. That can be done in the same vein as any traditional monoclonal antibody can be done. But then you have to the net, that's fine, and you got to look at drug tolerance. All of that has to be done in phase 1. It all has to be validated. You got to validate all 3 tiers: the screening, confirming, and titering methods. It's after you've gotten that validated, you really, or developed, you have to start looking at what's called domain specificity. And that's when you need different building blocks, is how I think of it. You need different reagents. You really have to start to understand, do I have antibodies against the linker? Do I have any antibodies against the payload? Do I have antibodies against the framework? And so really what you kind of need is a naked antibody. You're going to need the linker. And in some instances, very rarely do you have to look at the payload as a domain specificity. I wouldn't bother with that, again, unless I had some really high risk ADC, which again, I would engineer it differently to avoid that. But what I mean by domain specificity is you have to start to characterize the the actual the anti-drug antibody response. And how that's done is during the confirming step. So a screening method tells you, is there any antibodies against my drug? A confirmation step is like an inhibitory, where you saturate the method with drug. And if the signal goes down, it's inhibited, and it tells you, okay, that's confirmed as positive. Well, instead of using the total ADC, that's when you got to use just naked antibody. You can use some domain, the linker itself, if that inhibits. Sometimes there's fragments of the linker that are going to be cleavable linker. So it just depends on what those fragments might be. So some of our clients will make 6, 7 different versions of the linker and what it might be bound to, as well as even the total antibody pieces. You can start to try to discern if there's some portion of the antibody that's specific to that. So I don't know if that's making any sense, but basically the confirmatory assay starts to become like, that gets expanded into domain specificity. And instead of inhibiting with the total antibody, you start inhibiting with the linker payload, right? And you can start to discern okay, so it's confirmed as positive, but wait, hey, the linker didn't confirm positive. The linker payload didn't confirm positive. Okay, so I know it's the antibody framework.
Yes, absolutely.
Yeah, so it's just like all along the way, John, what it means is more validations, more of an investment in the immunogenicity. So I hope I covered that well. I felt like I stumbled my way through it, but we are all the time doing ADC immunogenicity assays, PK assays, and John, pound for pound, I think we're the best lab in the world at doing this.
Well, not that we're biased, but yes, I tend to agree.
I hope the competition's listening. I hope our sponsors are listening. Come challenge us. We'll take you on every time, won't we?
We love doing them because they're a fascinating bioanalytical challenge. And
well, John, even the 3 years we've been doing this podcast, we've been talking about it. But for 5 years, we've been really targeting ADCs, and really longer than that, I'd say for about 6 or 7 that I've been here, ADCs have been a prominent piece of our business line, and obviously one that is expanding really every quarter. It's fascinating how blessed we are to work with some of the ADCs that are out there, the next wave of them, and how powerful they are. And obviously, a couple on the market that we've solved some really extensive immunogenicity problems, because they're tricky, John. This is especially when you talk about payloads that fall off, and it seems like the more effective a drug is, the harder it is bioanalytically. It doesn't matter if it's a small molecule, a large molecule, an ADC, or whatever it is. The more effective a drug is, the harder it is bioanalytically, John. So,
yeah, it's been good for us, because when we talked about the new technologies coming through, we're actively supporting programs that involve most of those. So you're seeing where the field is going, and yeah, like you say, bioanalytically, it's certainly not standing still. So I think, shall we wrap up and then?
Yeah. Tell me about your weekend plans, John. What's going on?
My weekend plans are really more sort of long term, because we are under a month to my daughter's wedding. So it's all the final pieces are falling into place in terms of catering, music, venues, etc. But we are this weekend, because we'll have people here the day after the wedding, We have got a dumpster being delivered on Friday, so my weekend will be filling that, just generally tidying the yard, ready for people to and also there's been things like we've ordered a lot more outdoor chairs, so assembling those. You
have like a manual labor weekend. I'm happy for your wedding and all that, but that sounds miserable, John.
Well, it'll be fine.
On Monday morning.
To be honest, we've had, I don't know what it's been like in Kansas. We've had a very wet year, so all the plants are exploding in the backyard, which means it's not just the plants you put there intentionally. All the weeds are exploding. And it's got, because we've been traveling, it's got away with us. So we're actually, there's a gardening company coming in next week to try and clean up a lot of the stuff we don't want, and again be part of that process to make it much better for my day.
Yeah, we have had the most ideal weather for growth of weeds and grass. I mean, it's been cool in my 16 years here. This is far and away the coolest summer we've ever had. The weather is easily 10 degrees cooler than it normally is. That doesn't mean it's hot. I'm talking about low 90s instead of high 90s, John. And a lot of humidity we've got, but it's been rainy. Everything is so green. It is, if you're out here golfing and you get in the rough, it's a true penalty, John. I mean, I can't begin to tell you how hard it is to hit out of the rough here, but I digress a little. No, we've had a very, I don't know if it's just been it's not that it's been excessively wet, but we've had a wet summer. But couple it with it being cooler, and things are just, sometimes we're brown this time of year. It is green everywhere, John. So I use the rough. I half joke, but usually this time of year, hitting out of the rough is real easy. Right now, you go into the rough, you're losing a stroke because that ball's buried.
It's the humidity's been tough this year, I have to say.
My weekend plans are much better than yours, John. Although I guess it has a lot of rewards to it, but all of that sounded like work to me. I am celebrating my 22nd anniversary on Friday.
Nice.
Friday, I'm taking Friday off, and the wife and I we're going to our favorite Italian restaurant. It's called Jasper's. It's a family-owned restaurant here. It's about as famous a restaurant as you can get. The owner is nationally kind of recognized, and Jasper's is the last name. And they've got they this time of year they make mozzarella right at the table. They use Wisconsin curds, and they make it right in front of you. They knead it, cut it with some heirloom tomatoes. He's got these fresh tomatoes he brings in, so I'm looking forward to that little Saturday afternoon. We're doing that, and then we got some plans to tool around at some of our local, there's a place called Weston, which is like a half hour away, and Excelsior Springs is another option where you can go and, kind of go in some hot baths and stuff, like natural baths up there, springs up there to kind of relax. So we'll do something fun there. And if it all goes well, sometimes we like to swing by a casino and have some fun there. So that's my weekend. I definitely have some plans to cook. I'm always making something good I did can some pasta sauce, so I can't go without saying some pizza in my future. I did buy some dough this past weekend, so I might use my Uni grill, but I got a 3-day weekend, John, and my wife and I, we're not flying to Vegas or anything fun, but we'll tool around and really get out and have a good time and create some memories.
Yeah, so I got a pasta machine for my birthday from my doctor, so I'm going to start getting in the mode of making pasta and seeing how that works.
If you figure it out, John, let me know, because mine I've used mine like 3 times. I got one for Christmas. Every time I use it, it just comes out like sludge. It does not. So if you figure out, and I got a real nice one, but it just, I've used it like all 3 times. The 3rd time, it just feels like I'm eating like a manicotti instead of, pasta. Yep. So if you figure that out, you give me a holler. But
I think the next few weeks are going to be wedding focused. After that, I'll definitely be on the seeing what I can do in terms of pasta.
I've certainly mastered the egg. Yeah, I think we're good, John. Enjoy the rest of the week. Sorry, go ahead.
Yeah, I'm going to say exactly the same thing. I think it's time to wrap up. Enjoy your weekend and week, and we'll talk soon.
All right, we'll see you in the funny pages.